Okay, now on to the exciting topic of protein synthesis. Protein synthesis itself. So we've established several things as we have talked about our cell. One, all cells, well, not all cells. So most cells have this thing called the nucleus. Okay. Inside of the nucleus, we have these things called chromosomes. Okay. And in humans, there are 23 pair of chromosomes. Right? And all a chromosome is remember, it is DNA wrapped around histones, and they're tightly packed. Okay, and this DNA contains the genetic code. Okay, and we have a gene that codes for a protein. All right. So now what we're going to do is we're going to talk about how a gene gets activated. So if DNA is the blueprint, we need to find a way to build something based off that blueprint. Okay? So we already know inside the nucleus that we have DNA, and it's bonded. It's a double-stranded helix. It's a double-stranded helix itself. But it's not DNA. DNA can't travel outside of the nucleus. Okay? And proteins are synthesized in the cytosol. And if DNA can't get out of the cytosol, what we have to do is we have to have a way in order to get the diagram for the protein from the nucleus into the cytosol, where the ribosomes are. And that is the role of messenger RNA or mRNA. So when the cell gets a signal to make a protein, okay, and this is important, the cell has to get the signal to make the protein. There's this enzyme, step 1 in protein synthesis, 1, we have to make the RNA. And there's an enzyme called RNA polymerase. Okay? And what RNA polymerase is going to do is it's going to split open this DNA. Okay, just like the replication fork almost. Almost just like the replication fork. T-A-N-G-C-T. Okay. And so RNA polymerase, I'm going to do it in red. So here's RNA polymerase. It's going to read this. RNA pol is short for RNA polymerase. And we're going to read this. And RNA polymerase is going to read this. And it's going to build the mRNA based off of this template. So it's going to be A. Since there's no T, there's no thymine in RNA, it's going to be A, U, U, C, G, A. And that's what our messenger RNA is going to look like based off of this template, based on the bottom template. So now we have an RNA that looks like A, U, U, C, G, A. This is our mRNA. Okay, and it comes based off of a DNA template. Okay? All right, and that's the first step in protein synthesis. And then the next step, once you make mRNA, and they're very long, and there's a lot of nonsense in it. Okay, there are these other enzymes, and you don't have to know this, that edit it. They basically take out the nonsense, so it shortens the mRNA. Okay, so then here, so we're going to draw another cell right here. So here's another cell. Here's the nucleus. And inside we got this newly formed mRNA. And what this newly formed mRNA is going to do, it's going to go out into the cytoplasm. It's going to go out into the cytoplasm. Okay, and so step 2 of protein synthesis is mRNA moves to the cytoplasm. And that's the second step. And then step 3 involves ribosomes. Ribosomes bind the mRNA and they form the amino acid chain. Okay, so basically remember a ribosome has got 2 parts. Basically it's got a small part and a large part. And what's going to happen is the mRNA is going to be fed through here almost like ticker tape. And you can actually see this if you see it on an electron microscope. And I'll see if I can find some pictures for you in lab. Okay, and remember from earlier lectures that 3 codons code for a single amino acid. So in this case, let's just say, let's use our AUU. This is AUU, and the next one is CGA. CGA. And what that is going to do is that's going to get a transfer RNA, and that's going to have an amino acid. Sorry. Let's do it like this. A transfer RNA, and it kind of looks like a T, is going to have an amino acid attached to it. And this and this transfer RNA, guess what? It will line up with the mRNA. So it'll be UAA. So that UAA, what it's going to do is it's going to deposit that amino acid onto the ribosome. And the ribosome is going to hold on to it. And then I'm going to have another transfer RNA. And it's going to be, so GCU. And it's going to have another amino acid attached to it. And it will bind to that amino acid. So eventually what's going to come out of this ribosome is amino acid attached to amino acid. And it's going to be based off of the messenger RNA. The messenger RNA. Okay? And that's all it is. That's all it is. So the ribosomes are going to bind to the messenger RNA, and the transfer RNA is going to bring in the amino acids, and the ribosome is going to take those amino acids and just make a chain out of them. And what dictates the order of the amino acids? The messenger RNA. What dictates the messenger RNA? The DNA sequence. Okay, so here's what I'm going to want you to be able to do on an exam. Okay, I'm going to say, here is a DNA strand. Okay, here's a DNA. I'm going to give you a DNA strand. Okay, and it will be any like. Okay, it'll just be a bunch of letters, and I'll set it up into 3. So you'll always know what it is. I will ask you, what is the mRNA strand? What will be the mRNA strand? So you'll have to give me the mRNA strand. Okay, and then what I will ask you is, what is the amino acid order? What is the amino acid order? Okay. And so to figure out the amino acid order, what we're going to do is we're going to get, let me find it, an amino acid chart. Okay, we're going to get an amino acid chart. All right. So if you have it's not letting me write. Okay, so if you have Let me see if I can just put this into my OneNote. I think that would make it easiest. There we go. Okay, so if I have an mRNA, so if I have an mRNA, and let's say my mRNA is, oh, let me just start with the DNA. Let's say we have our DNA strand, and it's AUG. DNA strand is AUG. Then I say, okay, now what is the mRNA that comes from AUG? It'd be UAC. Now, what is the amino acid? What is that amino acid? So then what you do is you just look at this messenger RNA right there, and you say, okay, there's an mRNA. The first letter, so you take a look at this first letter right here. That's number 1. It's U, so it's going to be right here. Okay, so it's going to be in this. It's going to be in this row. Then you look at the second letter. It is A. Okay, so it's going to be in this row. It's going to be in this column. That row and that column. Okay, and so then what you do is you just look at the last letter, C. So then you can say C. Okay, so this would be tyrosine. It is UAC. UAC is tyrosine. Okay, so that would be TYR. 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 Okay, so we'll do another one. So the DNA, let's just say it is TTT. The messenger RNA then is A. Okay, so the first letter is A, second letter is A. So here, so it's going to be in this box right here. And then the third letter is A. So AAA is LYS. So the amino acid is lysine, LYS. Okay, so that's all I'll have you do. I mean, that will be the extent. That will be the extent of protein synthesis for me. It's not going to be super duper complex. Okay, so the last thing I want to leave you with as we talk about protein synthesis. Okay, so here is the general. Here is the general outline of basic biochemistry. Okay, this is how it actually goes out. Okay, and so the two terms that I want you to remember this. Okay, so here we got a circle here, DNA. This is DNA synthesis. Okay, going from DNA to messenger RNA. Okay, this is called transcription. Okay, that is transcription. Going from messenger RNA to a protein, that is called translation. That is called translation. Okay, and that's all I want to leave you with. That's all I want to leave you with protein synthesis. It's really not that difficult, and I'm not going to make it that difficult on any exam. So there we have it. The last thing we're going to do is we're going to talk about the cell cycle.